All right, hello everyone and good morning. Thank you for joining the Medical Microbiology and Infectious Diseases Bioinformatics Workshop, Introduction to Bacterial Genomics, and today we're going to focus on reference databases and taxonomic classification. So my name is Jill Rumore. I'm a Ph.D. candidate in the Department of Medical Microbiology and Infectious Diseases, and also the co-coordinator of the workshop series. So just a bit of housekeeping information. Just to let everyone know, all workshops are being recorded and posted to the MMID Bioinformatics Workshop YouTube channel. And of course, for the live Q&A, please feel free to go to Slido and use the participant code that's listed in the slide. As well, you can also post your questions in the chat, and we have multiple people in the room monitoring, so we'll be able to answer those quite quickly. Just a recap of the Bioinformatics Workshop schedule that has happened so far to date. So we have 4 workshops left. So the next 2 weeks are going to be taught by Taylor, and essentially we'll be providing an introduction to phylogenetic analysis, as well as tree visualization. And then, of course, the last 2 sessions are going to be open to the public, where this gives you an opportunity to come bring your own data sets, or if you had any issues with previous workshops, this is a great opportunity to ask questions and get maybe some of those tools running that you might have had challenges with. I would highly recommend that if you are planning to attend the April 20th and April 27th sessions, that you do come in person. It's much easier to troubleshoot in person rather than trying to do it over Zoom. For those in the room, I'm sure everyone's familiar with how to connect to the Wi-Fi. Again, just making sure you're using the appropriate login credentials. Okay, so for today, I'm basically going to be providing a basic overview of taxonomic classification and, of course, the importance of well-constructed reference databases. So we have a number of learning objectives for today, which are all listed in the slides. So we're going to briefly touch on what is taxonomic classification, what are reference databases, some of the current challenges with reference databases, as well as how to choose classification software, because there are a lot of programs out there. And then towards the end of the workshop, we're going to do a little bit of hands-on work where we're going to perform taxonomic classification on a publicly available data set using Kraken 2, and then time permitting, hopefully we'll have a chance to visualize those results in Pavian. So just to remind everyone, we're going to be using the same publicly available data set that we had used for last week's workshop. Now, I realize there may have been some challenges with running some of the tools last week, so we have uploaded the data sets that we're going to be working with today to the MMID Bioinformatics Workshop GitHub. So if you weren't able to get through the exercises last week, that's okay. The data sets are available for you today. Now, with that being said, although the data sets are not large, reference database is. So there was a bit of an expectation that you would have had this information downloaded prior to coming to the workshop, because it does take about 20 minutes to download. So it could lead to potential delays in participating in the workshop demonstration at the end of the session, or towards the end of the session. Now, if you haven't had a chance to download it, that's okay. I would recommend you get started on doing that now. So there are multiple links posted in the chat about step-by-step guide on how to download the data sets as well as the database. And I would encourage you to open those and follow along. If you do have any questions, we have multiple people in the room who will be able to answer you and help you with any questions you may have when trying to download either the data sets or the reference database. Okay, so what is taxonomic classification? So really, the ultimate goal is to try and determine from what organism did this read originate. And that's done for all reads in the data set. Now, the choice of assembly-based analysis versus direct taxonomic classification of reads will, of course, depend on the research question. However, mapping short reads directly to a reference database is a more sensitive approach and often the preferred method for taxonomic classification, and will be the focus of today's workshop. Now, this is just a very simplified figure depicting the typical analysis steps from raw sequence data to taxonomic classification of reads and ultimately pathogen detection or verification. Now, to perform read classification, we need 3 key elements: an appropriate tool, a reference database, whether that be pre-computed or custom built, and then of course some analysis parameters. Now, the reference database really is an essential component of the classification process as it contains data from previously sequenced microbial genomes against which the filtered sequence data is screened, and therefore it has a direct impact on the answers that we get. Now, all classification software requires a pre-computed reference database that, of course, is based on previously sequenced microbial content. Now, while most software is or commonly distributed with pre-computed reference databases, however, the content of these databases tends to vary quite substantially. However, one of the common things is that most of the data actually comes from the NCBI RefSeq complete genomes for microbial sequences, as well as the BLAST or NT or NR databases, which are for the high quality nucleotide and protein sequences. Now, a lot of the tools also allow the user to build a custom database based on your desired content or whatever your research question is. Now, this can be actually quite computationally intensive process, but it affords greater control over the analysis. So as I mentioned, the database really has a direct impact on the answers we get. So the quality and composition of the reference database is very, very important. So more specifically, contamination and incompleteness in the reference databases can lead to both false positives and false negatives. So a number of studies have been published over the years showing contamination in the public domain, particularly for draft genomes. So just I have some examples listed or shown in the slide. I'll get my pointer here. Just here. So in 2011, Longo et al. Identified 492 non-primate public databases from NCBI that contain human genome sequences. And then 2014 study found that portions of the genome from Neisseria gonorrhea actually belong to the cow and sheep genomes. And then we had another large-scale analysis in 2015 that showed that over 18,000 microbial isolate genome sequences were contaminated with the Phi-X bacteriophage, which is used as a control for Illumina sequencing. And then most recently, 2019 study that looked at the NCBI RefSeq database found that over 2,000 genomes were contaminated with human sequences. So it goes to show you there's just a lot of contamination out there in public sequence repositories. So beyond contamination, there's another issue of incompleteness. So sequencing efforts have primarily been directed or focused on human pathogens and other microbes of interest to humans, particularly those that we can actually culture in the lab. Now, there exists an enormous diversity of uncultured microbes that are often referred to as microbial dark matter. So what does this mean for taxonomic classification? Well, reads without a reference in the database will be labeled as either unknown or imprecisely assigned to the next closest taxon. So I mentioned earlier that a lot of the software that is available out there for taxonomic classification does come with pre-computed reference databases. And that's particularly, I'm showing here for the Kraken 2 and Kraken Unique software, which is hosted on Ben Langmead's GitHub. And today we are going to be using the standard database, which is 8 gigs. So one of the smallest Kraken 2 databases that I could find that was already built. So pre-computed databases can actually be very helpful because you don't have to go through the process or the trouble of building your own reference database. However, prior to conducting any type of taxonomic classification, it's extremely important to do your due diligence and evaluate and validate the reference databases. So you want to ask questions such as, does the reference database contain genomes for the pathogen you're interested in? Are the genomes of high quality? Who built the reference database? Is it a credible source? How big is the reference database? And do you actually have the computational resources necessary to run it? So we're just going to go through, I have 2 kind of case studies based on some of my experiences with reference databases. So here's a recent example of how a pre-computed reference database is not a one-size-fits-all. So for context, this work involved single isolate sequencing of various Vibrio species, and this was completed by one of our instructors, Taylor, as part of her research project. Now, when using the pre-computed reference database that was distributed with the Kraken 2 classification software, About 75% of the reads were being classified, meaning that around 25% were not. Now, initially, we thought these samples may have been contaminated, as anything more than 10% unclassified for sequence data that should have originated from a single isolate is quite high. However, Taylor, she did a very thorough QC assessment and all the samples, of course, checked out. So our next step was to investigate the contents of the reference database. And we actually found that a number of rare Vibrio species were missing. So to tackle this issue, we built a custom reference database that was enhanced for these Vibrio species and reran the analysis. And you can see here that when we reran it, about 95% of the reads were being classified or more, with 5% or less being unclassified. And this is what we typically would see for single isolate data. So taking a look at another example, so to minimize the likelihood of false positive classifications, the use of reference databases containing all domains of life, which is typically referred to as a standard or a universal reference database, is often recommended, even if your focus is on a specific taxonomic group. However, standard databases can be quite sizable. So they can get upwards of 100 gigs or more. And as a result, they do have high memory requirements, which may be out of reach for some users, so actually preventing you from conducting these analyses. So in a study that was recently conducted as part of my thesis work, we found that a universal reference database is not essential for accurate and reliable detection of, we focused on Listeria monocytogenes, which is a priority foodborne pathogen here in Canada. And what we did find was that you were able to achieve equivalent classification performance using a smaller or a foodborne pathogen specific reference database when the appropriate quality control measures, classification software, and analysis parameters are applied. Now, overall, this approach was less computationally demanding and more attainable for the broader scientific community. So you can see that when we applied the appropriate parameters, no matter what tool we used, we ended up getting the same answer. So that was great. So if you haven't already guessed it, really the take-home message here is always evaluate and validate your reference databases. Now, it's always good to have a benchmark data set or just a well-characterized data set where you know what is exactly in your sequence data. And of course, always including both positive and negative controls. Now, unfortunately, we don't have time to build our own database or the computational feasibility. But if this is something you might be pursuing in the future, I wanted to include some helpful tips that I have found to be extremely useful when when I've built my own databases. So the slide is a bit text heavy, but I did want to have all of this information in one place for easy referencing. So the first thing is, you always want to use complete or quality controlled reference genomes. And this will considerably help reduce the number of false positive classifications. And you can always screen your genomes using CheckM, which is a tool that we had used last week. And then you could take a look at the completeness and contamination, because as I had mentioned in the previous slides, just because a sequence is coming from NCBI RefSeq doesn't necessarily mean that it's always of good quality. So it's always important to double check and make sure that all of the sequences that you're putting into your database are the highest quality possible. The next thing you want to do is you always want to mask low complexity sequences. So these are essentially sequences that might be common across different taxa. So it can lead to non-specific mapping. Now, a lot of the tools will do this automatically for you when you're downloading the data from RefSeq. However, if you choose to use a software that does not offer this functionality, you can always accomplish this using Dust Masker. Now, we're not going to go over this today, but I have provided a link to the paper, as well as it's included in the BLAST suite of tools. So if you wanted to install it yourself, you would look for BLAST on Conda, and it would be part of that entire suite. Now, the next thing I recommend is that if you do have to use a draft genome, because I realize, that some of the pathogens or some of the microbes that we might be studying might not be available on RefSeq in complete form. So we might have to use draft genomes in our reference databases. So if that's something that you plan on doing, you want to make sure that you're filtering out contigs that are less than 1,000 base pairs. And this is because a recent study found that actually a majority of the contigs that are less than 1,000 base pairs were found to be contaminated with the human genome, among other organisms. So it's quite an interesting paper. I would recommend giving that a read. Now, to do this, there are many ways, but one of the easiest ways you can do this is to use SeqKit. So that is a tool that is available on Conda that you can install. And basically, you would just select the size that you want to filter out, and you would supply it with your reference genomes. And then, of course, including the human genome. And why is that? Well, I mean, it really is the #1 contaminant in the lab. So including this in the reference database will actually help to improve your classification performance. And there have actually been a number of studies that have shown that by including the human genome in your reference database can actually reduce the number of false positive classifications. And then likewise, I also like to include the contaminant reference databases, which is UNIVAC and EMVAC. So these basically contain the adapter and primer sequences, as well as the sequencing control phi-X. And of course, including these in the database can also reduce false positive classifications. Again, Kraken 2 and Kraken Unique, you can download these automatically from their respective websites. But if you choose to use a different tool, I've supplied where the FTP sites for both of those contaminant reference databases. Okay, so now that we have hopefully a better understanding of reference databases and the potential challenges that come along with pre-computed reference databases, the next step is to decide what classification software to use. Now, there are a plethora of tools out there, and there have been a number of benchmarking studies, a few of which I've shown here in the slide. However, when attempting to choose a classification tool, I typically like to consider 3 major criteria. And the first is availability. So the tool should be freely available and, of course, open source. The second is usability. The tool should have good documentation describing how to install and properly use the tool, as well as developer contacts for troubleshooting. And then the last is adoption. So the tool should be widely used or show potential of being widely adopted in the future. And of course, these tools are routinely updated and actively maintained by developers. Now, based on this criteria and the results of a number of benchmarking studies, there often remains one suite of tools that reigns supreme, and that's, I'm sure many of you have probably heard of it, and that is the Kraken software, which has been developed by the Salzburg lab at Johns Hopkins. So currently, there are 3 major versions of the Kraken software. So Kraken, Kraken 2, Kraken Unique. Now, in the slide, I've highlighted some key features of each. However, I would encourage you to read the publications that I've listed in the slide just to get a more detailed overview of each of the tools. Now, most importantly, the original Kraken software, sometimes referred to as Kraken 1, is no longer supported. So if you choose to use Kraken, I would encourage, if you are using Kraken currently, I would encourage you to switch to either Kraken 2 or Kraken Unique. Now, Kraken 2 is basically a more memory efficient version of Kraken, and it actually reduces the memory usage by about 85%. And this is because it uses much, much smaller reference databases, and that's why we're going to be using it today. So it runs about 5 times faster than Kraken Unique, which is more similar to the original Kraken software. However, with using smaller databases comes the possibility of more false positives, though it is quite minimal with Kraken 2. And with adjustment of some analysis parameters, those can be easily minimized even further. Now, unfortunately, with Kraken 2, the original Kraken databases are not compatible. So if you choose to use Kraken 2, you will either have to use a pre-computed database or build your own. Now, one of the interesting things about Kraken 2 is they've recently incorporated a feature referred to as unique k-mers. They sometimes refer to it as distinct minimizers, but in a recent paper that I looked at just a couple of days ago, they now refer to it as unique k-mers. So this is a feature that's been leveraged from Kraken Unique that actually has been shown to be more informative for pathogen detection compared to reads. And we'll talk about that a little bit more in the coming slides. Now, if you choose to, unfortunately, this is not generated by default. So if you did want to get the unique k-mer counts, you would have to include the report minimizer data flag in the script when you're running Kraken 2. And that's what we're going to be doing today at the end of the workshop. Now, Kraken Unique, that is another great tool. It is compatible with the original Kraken databases. However, the databases are quite large, and as a result, it is quite memory intensive. But of course, with the larger reference databases, the classification performance is a little bit better than Kraken 2. And again, this tool also reports unique k-mers. So we're going to talk a little bit about k-mers because I've been mentioning it a little bit, and I want to make sure that everyone has a basic understanding of what k-mer is, as well as a k-mer-based classification approach. So the classification approach for Kraken 2 is based on exact k-mer matching using the lowest common ancestor, or LCA for short. Now, this is one of the more popular approaches, as LCA drastically reduces the risk of false positive. However, it usually spreads the taxonomic level of the classification from the more specific to the more general. Now, for those who may not be familiar with k-mers, so they represent short sequences of a defined length. Now, for example, in this figure, the read is broken down into k-mers that are 4 nucleotides in length. Now, when conducting taxonomic classification, the ultimate goal, as I'd mentioned earlier, is to try and determine from what organism did this read originate. And we want to do that for all reads in the data set. So to accomplish this task, each read is broken down into its k-mers. And of course, the size will depend on the tool that you are using. And then it's compared to k-mers that are stored in the reference database to identify its most probable taxonomic label. Now, once all the k-mers have been searched for in the database, the read is classified according to the root to leaf path with maximal weight. So that just basically means that the read will be assigned to the taxonomic label with the most k-mer evidence. Now, since most tools use the lowest common ancestor, I just want to mention that if a read contains k-mers that are shared equally between 2 taxa, the read will be labeled at the higher taxonomic level. So for example, if a read matches both the turtle and the snake, then the read would be labeled at a higher level, for example, reptile. Now, in this example, if we follow the path from the root to the leaf, we can see here that this read most likely originated from a snake based on the k-mer evidence. So now that we have a better idea of what k-mers are, I wanted to revisit the concept of unique k-mers and how they can be useful for pathogen detection. So in this slide, I have 2 examples depicting read counts versus unique k-mer counts. And that's for 2 data sets, which were both obtained from the Kraken Unique paper. Now, if we take a look at the top figure, although the number of reads is quite low, we can see here that the number of unique k-mers is rather high. So this would suggest that each read matches a different location in the genome. Now, when we do a realignment of the map reads to the reference genomes, we can see that these reads are actually quite evenly distributed across the reference genome, suggesting the pathogen is likely to be present and therefore a true positive result. Now, if we take a look at the bottom figure, we can see here we have a higher number of reads and a lower number of k-mers, unique k-mers. Now, this would suggest a possible false positive identification, as these reads are more likely to be concentrated in a single location or very few locations in the genome, which we can see here looking at the realignment that we do. The reads are just piling up in a few areas. Now, as a result, we would consider this as a false positive result. So you can really think of unique k-mers as a proxy for genome coverage. Now, this feature is really exciting. It's something that I use in my own work, and I think it's very promising for pathogen detection. However, it's still experimental, and it does require further validation with well-defined negative controls, of course, to determine the minimum number of unique k-mers that would be necessary to support a positive pathogen detection result. And of course, this is going to probably vary for every pathogen. So there's still a lot of work to be done, but again, a really great feature that has been incorporated into the tool. Okay, so talking a little bit about Kraken 2 databases. Basically, they're made up of essentially 3 files, but unfortunately, none of them are human readable. Other files also might be present as part of the database build process when you download them, especially if pre-computed, but those can be removed if desired. So if you're running low on storage, as long as you have these 3 files, you're good to go. Now, one of the things about pre-computed databases is they don't always come with the information about what they contain, but there's a way we can work around that. So we can actually investigate the database ourselves when using Kraken 2 software by running a simple script. So that's the Kraken 2 inspect script, and that allows you to gain information about the contents of the database, and that will summarize it in a text file for you. So for example, today we're looking for Vibrio parahemolyticus. That would be something we'd want to check our database for. Does it actually contain Vibrio as well as Vibrio parahemolyticus? So it's always a good first check, especially if they do not provide information regarding the contents of the database. Now we'll take a look at the output files. So Kraken 2 generates 2 output files. Now, unfortunately, neither of them come with headers, and this is something that they have never included in the tool, which I'm not sure why, but it has been, of course, something that would be extremely useful. But nevertheless, I have included the headers in the slide to provide some clarity when we're going through the 2 outputs. Okay, so the first one is the standard output, which I refer to as the read classification. So it basically contains 5 tab delimited fields. So the first one indicates whether or not the read was classified or unclassified, so C representing classified. The second column is the sequence ID, or the read ID. The third is the taxonomic ID, so whether or not that read was actually classified. So this read was classified at tax ID 670, which I believe is Vibrio parahemolyticus. So that's great. If it wasn't classified, it would appear as a 0. And of course, you would see a U here as well. And then here we have in the 4th column, that is the length of the sequence in base pairs. And because we have paired end data, we're going to see our sequence length for both our forward and reverse reads. Okay, and they're just separated by the pipe. And then 5, the 5th column will basically be a space delimited list indicating the lowest common ancestor mapping of each k-mer. So what does that mean? Well, if we take a look at this bottom one here, what that basically means is that the first 4 k-mers are mapping to tax ID 670. The next 54 k-mers in that particular read are not mapping to anything. The next 8 k-mers are again mapping to tax ID 670. And then this particular character indicates a separation between your forward and reverse reads. And then again, the same thing. We see the first 4 k-mers mapping to tax ID 670. Then we have our 59 k-mers not mapping to anything. And then our last 3 k-mers mapping to tax ID 670. Okay, so taking a look at the report file, and this one is usually of the most interest to individuals because it will give you your read classification information. So the Kraken report file outputs lines containing 6 tab delimited fields by default. However, if we run the report minimizer data flag, we will get 8 fields. And this is what I'm showing in the slide here because that's what we will be doing today. So the first column corresponds to the percentage of fragments that are covered by the clade rooted at the taxon. The second column represents the number of fragments covered by the clade, or sorry, the second column represents the number of reads rooted at this particular taxon. The third represents the number of fragments directly related to the taxa. And then 4 represents the number of minimizers that are found to be associated with the taxon. So these would be k-mers. And then we have our unique k-mers, which is the estimate of the number of distinct or unique k-mers that are associated with the particular taxon. And then we have our rank. So for example, we have here S for species, Vibrio parahemolyticus. And then we have our NCBI taxonomic ID, and then our indented scientific name. Now, for more information about these output files and to get sort of a more thorough understanding of how they work, I would highly encourage you to visit the Kraken 2 manual, which is listed in the helpful resources at the end of this. It's also listed at the bottom of the slide, but also in the helpful resources at the end of the workshop. Okay, so I guess everything went a little bit quicker than I expected, but this will give us some time to run some of the tools. So before we jump into that, were there any issues with downloading the database? If you had any issues, you can pop that into the chat. Okay, I'm getting some thumbs up in the room. Looks like everyone may have had a chance to download it. Okay, so we are going to get started. We're going to run Kraken 2. Okay, so the first thing we're going to do is we're going to open up our terminal. Okay, and we're going to make sure that we are in our reference databases directory. So I will go back a few. So if everyone remembers, if you could navigate to your conda workshop directory, so that'll likely be in your mount, C, users, then your username, desktop, and conda workshop. And remember, it's always CD. So I'll just put it in here so everyone can see. Okay. Give everyone an opportunity to get to that folder. Okay. Okay, so once you're there, then you want to make sure that you have the reference databases directory. So to do that, we can do a list. So if you haven't created it, you won't have it, but I already have. If you need to create the directory, we're going to use the mkdir or make directory command, and we're going to type in reference databases. And then you're just going to hit enter. Okay. But this is where you're going to find your data sets that were previously downloaded, as well as the Kraken 2 database. So now we're going to move into that directory. So we're going to CD into reference databases, and we're going to press enter. And then we're going to list the contents of the directory by typing in ls and enter. So let's give everyone a moment. So in your directory, all you should really see right now, you won't see the data sets full, or you may see it if you followed the instructions, the pre-workshop instructions. So you'll for sure have a zip file, and hopefully you'll have the Kraken 2 database. If you've unzipped it or decompressed it, I should say, you'll have this directory as well, Kraken 2 database. And then if you've also decompressed the data sets, you will have the data sets folder. Now, if you haven't, so what we're going to do is we'll unzip the data sets. I won't do that because I've already done it, but this is how you would decompress the data sets folder by running unzip and then press enter. And that shouldn't take long at all to decompress. The samples for today's workshop have been downsampled, which was indicated on the MMID Bioinformatics GitHub to about 20 times coverage. So they're a little bit easier to work with today. Okay. So once you have that unzipped, and then of course, we want to make sure that your database file is already also decompressed. Now, if you haven't done this beforehand, this is going to take some time. Although, we are a little bit ahead of schedule today. But if you do need to decompress your standard reference database, you would just type in tar-xbf followed by Kraken. And then you could probably tab complete it once you have the standard database. And then you would press enter. Now I've already decompressed my database, so I won't be doing that. But if that is something that you need to do, this is the code that you would run. Okay. And again, that will take some time. I think when I was doing some testing, it takes about around 20 minutes. So you have to be patient. Unfortunately, there's just no way of getting around that for running taxonomic classification software. And this is actually one of the smaller databases that are available. Okay, so the next thing what we're going to do is now we've decompressed all of our files. What we're going to do is we're going to move into the downsampled host remove fastq directory, which is located in our datasets directory. So we're going to do that by typing in cd datasets. You can always tab complete. Okay, and then we're going to move into the downsampled. And we can always tab complete. So you can type in down and then tab complete. And we're going to press enter. And then, of course, once we're in the directory, always want to list the contents and see what we have. Now, yours might be gzipped. If they are gzipped, what you can do to decompress those, we're going to run gunzip. I've already decompressed mine, but if you see the .gz ending, you will have to run this. And then we're going to use the wildcard character because we want to unzip or ungzip all of our files, and then the .gz extension, and you'd press enter. And again, that won't take too much time because the files are all relatively small. Okay, so once we've verified that our FASTQ files are there, what we're going to do is now we're going to return to the reference databases directory. So we're going to do that by changing directory, and we're going to move up 2 directories. So just by cd .. followed by another .. We're going to hit enter. Okay. So now we can see that we've returned to our reference databases directory. Now in this directory, we're going to make a new directory called Kraken to output. I've already made mine, but the command to do that is mkdir, so make directory, and then we're going to go Kraken to _output. And this is where we're going to direct all of our output when we're running Kraken 2 in the, I guess, the next couple of minutes here. Okay, and then you press enter. Now, of course, once you make a directory, I always like to double check and make sure that it's actually been created. So I will type ls to make sure, so list the contents of the directory, and I can see here that I have my Kraken 2 output, which is great. So now I'm ready to run Kraken 2. Well, not quite. So first we're going to activate our conda environment containing Kraken 2. So that would have been installed, I think, in week 2 of the workshops in your conda workshop environment. So to activate our conda environment, we're going to go conda activate, and then we want to activate that conda workshop environment. And we're going to press enter. So now we can see here, we are in the conda workshop environment, and it appears beside our name. Now, just to double check that we have Kraken 2 installed, we're going to list the contents of the environment by typing conda list. So you're probably going to have a lot of programs in here because we've installed a lot over the past few weeks. We're just going to want to scroll to where, because it's all alphabetized. And we can see here we have Kraken 2, perfect, from Bioconda, and it's the right version. Everything looks good. Now I'm just going to pause for a moment before we move on. If there are any questions or if anyone's having any difficulties up until this point, feel free to put them in the chat. Okay, getting some thumbs up in the room. Okay, glad everyone is able to follow along. Okay, so the next thing we're going to do is we're just going to check out the Kraken 2 man page. So this is going to give us a list of all of the possible options that we could use to run Kraken 2. Now, we're not going to go through all of them, but this is something that you should be doing before you run any tool that you're not familiar with. Take a look at the options, see what they do, because some of them might be quite helpful to your own research question. So to do that, we're going to type Kraken 2, and then we're going to go --help. And we're going to press enter. So we can see here, we have a list of different options on how to, as well as a usage statement, and then a list of options that we can use when performing taxonomic classification with Kraken 2. Now, the one option that we will be using today for sure is the report minimizer data. Okay, so we're going to run Kraken 2. So we're going to start by typing in Kraken 2, which is the main command. Now, next, we're going to point to our database. Okay, so if we remember, that's in the current directory. So /, and then we can type Kraken, tab complete. And then if we remember, it was the standard. Okay, and then we're going to, if you tab complete that again, you'll see two things come up. We have our directory, which contains our database, and then we have our compressed file, which we don't want. So we want to make sure that we're selecting our directory. Okay, so just add a / at the end. The next thing we're going to do is we're going to run it with 2 threads. Typically, if you have access to high performance computing, you'll likely want to run it with more. Of course, this is going to depend on your data set. So again, I want to remind everyone that if you are using the high performance computing cluster, that you are using the Slurm workload manager to submit all your jobs. And as well, just reviewing the conda workshop documents from last year's workshop series, just to familiarize yourself with optimizing the Slurm workload manager script to use the appropriate amount of memory when running these jobs. But for now, we're just going to use 2 threads. Okay. The next thing we're going to do is we're going to create our report file. Now that is where we're going to put in our Kraken to output directory. So that again is in this directory, so the current directory. And we're going to type in Kraken, and then it was called output. Oops, Kraken output. Okay. And then we got to give it a name. So the sample we're running today is the same one that we ran last week. So I will type it out, and then I will give everyone an opportunity just to type it out because it is a little long. Okay, I'll just pause for a second and let everyone type that. So it'll be sample 2368311. And then I always like to add the -K2 report file at the end, so I know that that corresponds to my report file. So the next thing we're going to do, as I'd mentioned, we're going to put in the report minimizer flag. And that is so we can get the unique k-mer information. And that's using report minimizer data. Okay. Now, because we have paired end data, we need to use the paired flag. So that will be next. And now we're going to point to each of our reads. So our R1 and our R2, which again is located in our data sets downsampled. So in the current directory, so the / dataset, so we can just DA, you can tab complete that. Okay. And then we know it's in downsampled, so we can type the first few letters, tab complete. And then we're going to type in the first few letters. And because there's only 2 FASTQ files, or I guess 2 samples in this particular folder, we can tab complete, and we know we want 2368311. So we'll just press a 1, tab complete. Our first read, we want R1, tab complete. Now we're going to do the exact same thing over again for the R2. So again, we're in our current directory. We're going into data sets, downsampled. And then again, we're now going to indicate the R2 for that same data set. Now, the next thing we want to do is we want to direct where our standard Kraken 2 output, so that's the read classification, where that's going to go, and that's going to go into our Kraken 2 output folder. So what I do is I just direct my output to my Kraken 2 _ output directory. And then I'm going to call it, be very similar to the report, except we're going to have a different extension. So we're going to type in our sample name. And then K2 read classification .tsv. So we'll just pause a moment and let everyone catch up. Okay, so once you have everything typed in, we're going to press enter, and we're going to see that as it's running, it's going to print information to the console or to the terminal. But in the meantime, we're going to go back to the slides, and then we'll return to the console. It should only take a few minutes to run. And again, if you'd like to try these demonstrations on your own with the remaining data set, of course, there is a step-by-step guide with all the code, as well as Grace has been super wonderful, and she has put all the code from today's workshop into a single document, which is available on the GitHub for this particular repository for this workshop. So you can easily just take that one file and you can have all the code easily accessible. So once you're done performing taxonomic classification, you can visualize the Kraken 2 reports, or really any report from the Kraken software using the Pavian Shiny app. And basically this particular program compiles all of the read counts per species and allows between sample visualization and comparison. Now, this tool wasn't necessarily developed for single isolate data. It's actually a tool that's primarily used for metagenomic data sets, but you still can visualize the data for single isolates. And it's kind of a nice way to easily to take a look at your data as opposed to looking at lines and lines in a spreadsheet. Now, one thing I want to point out is that typically Pavian would be installed in R. I didn't think we'd have enough time to do that, so that will not be covered today. However, they have really great documentation. This is one of the few labs, the Salzburg lab, that really spends a lot of time documenting all their tools so that people can easily run them. So if you would like to install Pavian in R, I would highly recommend that you visit the GitHub, and it's just a few simple lines of code to get the program working. Now, with installing Pavian in R, you have a little bit more flexibility, so you can increase the memory. So if you have large data sets, you're more easily able to upload that to Pavian, and it is much faster. So the program we're going to be using today is Pavian, but it's just from their locally hosted server. So it is a bit slow and doesn't have all of the functionalities, at least yesterday when I was testing it out, that you would normally have if you were to run Pavian from R. Okay, so we're going to run Pavian. So if everyone could open their browser, I'm going to go. Okay, I'm just going to reset. So the website is just listed here at the top. You can also copy and paste into your Internet Explorer, your internet browser, and then everyone should have this screen. So what we're going to do is we're going to just return to the terminal, and we're going to see if our stuff has finished running. And yeah, it did. So perfect. And we can see that most of our sequences were classified. And then, of course, we have a little bit that are unclassified, and that's to be expected. Okay, so that's great. So we can actually use that output to visualize in Pavian. So what we're going to do is we're going to click on Browse. Then we're going to go to our desktop, which is where we have all of, we have our Conda workshop directory, or wherever you have your Conda workshop directory saved. Okay, we're going to double click on our Conda workshop. We're going to go into reference databases. Now we know we put our Kraken 2 output in our Kraken 2 output directory. So double click on that. Okay, now you'll see here I have, oh, I must have typed in something wrong. So we can see here, so we don't really need the read classification file right now, but you would normally see a report file and a read classification file. So I'm just going to return to Kraken 2 output. So there may have just been a typo. So I'm just going to rerun that just to make sure. Okay, but in the meantime, what we can do is we can upload the report file. Or yeah, no, I do. Sorry, I'm looking at the wrong sample. So we do have both reclassification report file. So what we can do is you can upload the report file. So you'll click on this particular sample, but because I'm rerunning it, I'm going to select the other sample. So you'll click on the report file, and you'll click open. We'll see that it's loading. I'll maximize that. So we can see we've uploaded our sample successfully, our Kraken 2 report. Now, the next thing we can do is we're going to click on sample. And then we can visualize the Sankey diagram so we can see how our reads were classified. And most of them are classified as Vibrio parahemolyticus, which is what we want. If they were classified as anything other than that, that might be of concern because this is the data set that we were expecting. Now, there are other things you can do. So you can hover, should appear there. So, what you can do is if you wanted to compare data sets, we'll have to upload more than one. So I'm just going to go back and I'm going to upload a second report for you to see. So I'm going to go back and I'm going to upload both of my Kraken 2 reports. You'll only have one for now, but if you'd like to go back after the workshop and rerun the remaining sample, then you can put both of those in Pavian as well. So we're going to open those up. We're going to do the same thing. We're going to click on sample. We're going to look at our Sankey diagram now. When we hover, so this was working yesterday, but unfortunately, so what it will do here is it might still be running. That might be why. Nope. So when you hover over, I apologize, it's not working today, but when you hover over any one of these taxa, it will provide you with a bar graph here showing the number of reads that are identified at the specific level. And then it'll also show an orange bar that are identified at the children. So anything, for example, Vibrio, it'll show everything in turquoise and Vibrio, and then everything that was Vibrio species will be in orange. So it's kind of a nice little thing or a nice little feature to have if you want to compare samples. Again, it's particularly useful for metagenomic data sets. But again, if you want just a quick sort of visual of your single isolate data, it's always nice to view it in Pavian. Now, there's a lot of other options here. Unfortunately, in this particular version of the app, a lot of them weren't working yesterday and take a long time to process. However, if you do decide to install Pavian in R, this is functional, and it'll give you a nice breakdown, again, of the report, where you can visualize the number of reads as opposed to looking at it just in straight Excel. And again, there's a step-by-step guide on how to get those results into Pavian. So before we sign off today, above and beyond some of the links that I've included throughout the workshop, I also included a few extra resources that may be of use to you in the future. I would encourage you to visit them when you have time, especially this step-by-step protocol, which the Salzburg lab has just recently put out using the Kraken suite of tools. And they sort of give you a nice little step-by-step for classifying your data, complete with actually some quality control steps that we also did the previous week. And then again, too, there's some great information, additional information with respect to how to choose a classification software, particularly within the Kraken software suite. So just to close, I want to thank everyone for attending today. And I just want to also remind everyone, if you could just please take a few moments to fill out the exit survey, that would be really, really helpful. Your feedback is super valuable, not only to improving each of the instructors' workshop style or teaching style, but also the delivery of future workshops. So with respect to content and pacing. So that would be very helpful for us. And of course, it only takes a few minutes. I think there's only 3 questions. So that would be really wonderful. Thanks, everyone. We will see you next week. Take care.